to the conversation today by Kate, Ibrahim, and Beatrice, but in a different context. Development of supervised barrier is critical for ion partitioning between senescent and non-senescent tissues in a succulent halophyte Sarcocornea quinqueflora. This was a part of my PhD study under supervision of Professor Sergei Shabala and Associate Professor Lana Shabala. So senescence of plant tissue is an important developmental trait that enable an evergreen or a perennial to retrieve, recycle, and remobilize nutrients from elder to younger or upcoming seed tissues. Perennial plants seems to utilize the process to discard excess salt being accumulated in their outer tissues through salt shedding. One of these perennial plants is called Sarcocornea quinqueflora. Sarcocornea quinqueflora is a succulent halophyte. As you see here, the plant consists of assimilating oblong internodes, or we can call them as beads. The plant has an important anatomical structures. It has two distinct types of tissue, biological barriers, which is called endodermis-like layer, and additional photosynthetic layer located in between the stele and the endodermis-like layer. This was found in Sarcocornea fruticosa and Sarcocornea pernis, and we also found it when we take the anatomical structure of this plant. So we try to answer some specific questions regarding this plant. Does the development of the superized layer vary under elevated salt concentrations? How the superized barrier development may affect the mineral distribution over senescent and non-senescent tissue, along with the initiation of salt crystallization in the bead's outer tissues. What might be the role for the internal photosynthetic layer in the acclimation of these perennial halophytes under continuing saline exposure over successive seasons? Therefore, we collected Sarcocornea quinqueflora plant from the south arm coast of Tasmania, and we asexually propagated these plants from semi-hardwood cuttings. The well-rooted plants were transferred to their striking potting mix, and the uniform plants were grouped in 6 groups to be treated with their relevant salt concentrations ranging between 0 and 1,000 millimolar sodium chloride. 15 plants were used per group to ensure enough materials to be used in the upcoming analysis, which is morphological, anatomical, and physiological analysis. You can find much more about the plant material and the asexual propagation in our published paper earlier this year. We have taken our growth parameters after 10 weeks of treatments with salt, different salt concentrations, when the differences between all the levels become much more obvious, specifically with the higher salt concentration. If you can see here, 6 weeks of treatments doesn't show much more differences between these salt levels, but here it became more obvious. That's why our agronomical characteristics for Sarcocornea quinqueflora have been taken after 10 weeks. As you see here, all the agronomical characteristics show that Sarcocornea quinqueflora shows the classical response of a halophyte with a growth reduced under 0 millimolar and an optimal response between 200 and 400 millimolar sodium chloride. The growth also promoted until 600 millimolar, and after this, the growth declined again. These are many agronomical parameters such as shoot fresh weight, root fresh weight. The optimal response that we got was enhanced largely by root length and shoot length and leaf stem length, leaf stem branch length. Here you can see an individual leaf stem with a bead and leaf stem branch. Here is the main propagated roots and the main leafy stems. We found some interesting changes in the color of different individual leaf stems. Here you can see the development of the red color occurs only from 600 to 1000 millimolar sodium chloride. Also, you can see weathering happens. You can see the weathering happens here under severe salt concentrations. But here, weathering was so late and came 8 weeks later than developing here. The change in the color of the individual beads could be categorized in 3 routes. The first route at 0 salt dosage, where we didn't find any red color development, but the color slightly developed under the optimum salt dosage between 200 and 400, and the color became much more obvious under severe salt dosage ranging between 600 and 1000 millimolar. Also, you can see here salt crystals protruded only and was developed under optimum and severe salt. It means under salt treatment, but it was absent and doesn't develop under 0 salt concentrations. Therefore, when we take the anatomical dissecting of these dry locking beads, we found that salt crystals developed in the outer tissues under salt, in the presence of salts, but it was these salt crystals were absent in no salt treatments. We still have an interesting tissue, which is the endodermis-like plier tissue, enclosing living tissues. First analysis of SEM analysis of these crystals showed that they are mainly consisted of sodium and chloride, but this is the beads growing under 800 millimolar, but beads at no salt concentration doesn't show any of these bright fields. Before we show the changes happen in the anatomical structures, we can have a look here on the Sarcocornea quinqueflora bead anatomy. The plants have senescent or outer tissues, consist of epidermis, palisade tissue, and water storage layer. And then the plant have internal tissues, which will be maintained with the plant. It consists of endodermis and internal photosynthetic layer, and the stele area consists of 6 to 8 bundles. Here we can see 7. And here, this is the internal tissue in the primary growth. We can't see here secondary phloem and secondary xylem, but we can here see that the cambium ring developed here give rise to an outer secondary phloem and an inner secondary xylem. So the change, the anatomical change in leaf stem regions was followed in bottom, middle, and top leaf stem regions under different salt concentration. We followed the change focusing on 2 main issues. The first one is the development of the endodermis-like layer and also the secondary growth. Secondary growth can be followed through watching the cambium ring, which is decreasing with increasing salt, and then it completely disappears under high salt concentrations. Here we can see the endodermis-like layer increased in number of strata and thickness with increasing salt concentrations. So this is a number of endodermis-like strata, I counted them, and the increasing with increasing salt. We have tissue contribution to bead diameters. We measured different diameter of these tissues, and you can see here that the endodermis-like layer contribution is increasing with increasing salt from 0 to 1000, and here also the same. Then we measured the outer and inner concentration of minerals, sodium, potassium, and chloride, in outer senescent tissues and inner non-senescent tissues. And we find a very interesting results here. We found that all sodium and chloride accumulated largely in outer or senescent tissues, and they have lower concentration in the inner tissues. You can see here, this was reflected on the sodium-potassium ratio. Here, 16 comparable to 2.8. So,
just the outer. One minute.
We have the outer to inner. We think the outer to inner ratio of individual elements affect the senescent processing. That's why we provided a model depicting the strategy developed by this plant. The model relies upon two sources of energy and two sinks here. Here, source one fuel the sink one for an efficient salt sequestration in these cells. I just will go to the conclusion because and here we can see the development of the endodermis-like layer, and our conclusion. We suggested that the salt tolerance strategy in Sarcopenia quinqueflora builds up on two main features. The first one is accelerating the development of the biological barrier, and the second feature is a difference in the energy supply. You can find more on our recently published paper. Just yesterday, we got the final version of this paper. And also, you can find more about the importance of the biological barrier in relating to the plant anatomical structure. And you can find a clear link between the plant anatomical structure and the importance of the endodermis-like layer in terms of exposure to high ROS concentrations. Thank you very much. And sorry for any shortage, because this is my first oral presentation in a conference. I'm so confused, that's why probably. Thank you very much.
Any questions?
Oh, thanks, Hassan. That was a really nice talk. I just wonder, is Salicornia a CAM plant? Because I was just wondering how the
it's a perennial plant.
I mean the photosynthesis. Do you know if it's a CAM plant? I mean, I'm wondering how the carbon dioxide's getting past the endodermis into that second photosynthetic layer.
We have in between, because the anatomy of the internal photosynthetic layer seems to be unique. We have 2 or 3 cells just forming a bridge-like structure and living in between spaces. And I think this space is probably responsible for getting the air to these layers. Yeah.
Oh, it's beautiful anatomy. Thanks, good story.
Anyone else? I have a question.
Yes.
What caused that color change when you put it in like a higher salt concentration? Do you know? I'm just curious about the color change.
I didn't get it.
Oh, do you know what caused the color change in the plants when you put in higher concentration?
What caused that?
Yeah, like, do you have any idea? I'm just curious because it looked pretty. Yeah.
What chemicals?
Or, yeah.
Cause what?
The color change, like you showed.
The change in color?
Yes, yeah.
It's betalains, because here we are in the Amaranthacea, and Amaranthacea under the Caryophyllalis. And Caryophyllalis usually has plant families that form betalains instead of anthocyanins, because anthocyanins are normally responsible for the red color. So here we have betalains, because we are under Amaranthacea.
Yeah, thank you.
Thank you very much.